Welcome, everyone. We're going to get started, and we'll probably have some more people come and join. But thank you for attending today's webinar on Lymphomoids, a tissue-based ex vivo culture system for lymphoma therapy screening, a part of our webinar series of VitroGel Hydrogel for ex vivo tissue explant models. I am Christopher Oak from the Well Bioscience, and I will be your moderator alongside our speaker for today, Dr. John Wong from the Well Bioscience, and Dr. Albert Santamaria Martinez from EPFL. A few items before starting. During this webinar, you can utilize the Q&A tab on your left side for any questions you have. We will collect and present them during the Q&A session after the end of the webinar. We will have some quick polls segments during the webinar as well. Feel free to participate as we go along. Also, the webinar will be recorded and can be accessed later for your convenience. We will send those out to your registered email address after the webinar, and once it's available. Let's begin. I will introduce Dr. John Wong from the Well Bioscience. Dr. John Wong will start with short introductory on VitroGel and the application of usage. Afterwards, we will turn it over to Dr. Albert Santamaria Martinez to present their research on lymphomoids.
Okay, thank you, Chris. And welcome everyone to join us for this webinar. So before we jump to these very exciting lymphomoid models, I would like to go over a few slides about our VitroGel system, the platform we provide for this ex vivo model. So for the VitroGel system, it provides an optical xeno-free environment for the long-term culture and preserve the phenotype of the ex vivo tissue plant. With this model, we can use it as the patient-derived organoids, the patient-derived tissue, humanoids, and the culture them for days, weeks, or even months. So this is platforms that can preserve all the phenotypes and provide the reliable disease models or personalized medicine for drug screening purpose. A few key features that our VitroGel system provides here is the room temperature stays stable the platform, so everything says operate at room temperatures. It's the pure synthetic system, so you don't need to worry about any these animal compounds that are going to interact with your tissue sample. It's very versatile. We are not just using these platforms for ex vivo model. We use it from the 2D, 3D cell cultures, all the way from to the organoid cultures, stem cells that scale up, and even use it for the in vivo and injection. In terms of the injections, the VitroGel system has very unique injectability, so that it can maintain a long-term injectable properties. Even you mix the cells with the hydrogel, it's still forming a hydrogel form. The gel is still injectable by showing a very unique shear thinning and recovery properties. It's very easy for the cell harvesting, for the downstream analysis, and it's automation friendly. A quick comparison of the synthetic systems that we create and compare with the animal-based ECM. Besides the competition of the pure synthetic system, the preparation time for the operation is way shorter than the animal systems, which you need to cool all the pipette tips and others the system. It's the room temperature stable system, so all the protocol is performed at room temperature. And the other thing is that we are able to easily manipulate the mechanical strength of the hydrogel. In terms of some animal-based ECM, the high concentration hydrogel doesn't mean that you have a higher mechanical strength, mainly because that you have the higher protein concentration. But for our hydrogel, we are able to very easily to manipulate the mechanical strength of hydrogel by simply adjust the concentration, and that shows the very critical properties that for ex vivo models, when you need to adjust the microenvironments, the stiffness for the different tissue, and preserve the phenotype of the different tissue. Because it's a pure synthetic, so we have the very consistent of the batch to batch, and also the very easy cell harvesting with our enzyme-free cell harvesting solutions. We can harvest everything at 37, less than 20 minutes. Shipping at ambient temperature, and we don't need to waste a lot of animal to make this hydrogel system. And the way we are not just provides the hydrogel, actually we try to provide the end-to-end solution. So besides the hydrogel and then we have the ready-to-use version, which is like designed based on the application and the cell types. If you tell us that what application, what's the cell types you need, we can come out with the ready-to-use version. You just need to directly mix with your cells and use it from there. We also have a high concentration hydrogel, which is designed based on the hydrogel's properties. And we also have a bio-ink. So besides the gel products we develop for a 3D model, we also provide some spatial coating or like design of the well plate for 3D cell culture, hydrogel-based 3D cell culture. We also built this all together with the supplement system and make it as the research kit. And then we have the recovery solution, and eventually we have these downstream analysis kit. This is the one example. We have this Cytos 3D kit for like live data assay, and it's very easy to use. Can quickly determine like the whole tissue or the organoids penetrates into the organoid and showing like the live and dead cells and whole organoids. So it's a great tool for the drug screening. Today's like when we come out with these lymphomas models, that one major product we use is our high concentration hydrogel. And I think it's the one of these VitroGel RGT has been used for this system. So as I mentioned, the concept behind this high concentration hydrogel is designed based on the hydrogel properties. So the gels come with like a high concentration, so we are able to use the dilution solution come with the hydrogel to adjust the different mechanical strengths. Each hydrogel comes with the one single functional ligand modification. So if you want to understand that how the microenvironment, how the single functional ligand impact on your cell growth, you can use this hydrogel, take one cells and test it on the different microenvironment. Also, this is currently the only tool in the market. It's very easy for people to take a different formulation and mix and match to get what they want for their own microenvironment. So it's a powerful tool. As we always say, it's like a Lego block. It comes with the different shape, different colors, and you just use your imagination to create your own microenvironment. In terms of the application and then the protocol, we typically has the several different protocol, and for the tissues that embedding protocol, we have like 3D cell culture protocol here, which is the very easily that we can adjust the gel strength by mixing the hydrogel with the dilution solution and then directly mix with the cell suspension, adding them to a well plate and adding the medium directly on top. All what we can do is that create a thick layer of the hydrogel and sit the cells on top. So in that way, we are able to mix the gel and mix the cell medium directly, putting as the layer of the hydrogel without the cell, and then we add the cell directly on top. With that, actually scientists that can be very creative to using it as the sandwich methods or different methods combined. And for some of these tissues, explants or the ex vivo model, we use these combination models. We add the layer of the hydrogel at the bottom, we add the tissues, and then we add the hydrogel to embedding the whole tissue in this transwell. And in that way, you can embedding the tissues that's three-dimensionally, and because the size of the tissue, you don't need to worry about the tissues go to the bottom. You can create a layer of the hydrogel to protect the tissue from the bottom. We do have few publications regarding the different tissue model, including the breast, including the lymphoma models we're going to talk today, and some carotid model. But these platforms can use that for most of the tissue model for the ex vivo model, and our applications and then success case is increasing at daily basis. So we really welcome the people who want to use this platform for the ex vivo model for the long-term culture, preserve the phenotype, and use it for drug screening. And it's not just for the compound screening, it's also used for some immuno-oncological culture as well. With that, I want to emphasize that we always say start with the end in mind. The reasons that we create for these synthetic hydrogel is what we see is a lot of these great 3D technology can only stay in the lab based on this animal-based ECM. And we want to open the door for clinical application. We want to open the door for lab automation. With that, we need to think all these different details to really create a synthetic microenvironment, not just like putting the cell together, but also really the function, make it like the cell feel like they are home, so they can perform normally. And with that, we hope that we can really is that to push these 3D cell culture and 3D ex vivo model towards for the clinical application with our synthetic system. So we are working on our GMP grade products, and hopefully in these years we're going to push these ones for the real preclinical and clinical application. And with that, I want to get back to Chris. Thank you so much.
Thank you, John, for that quick introduction of VitroGel and how it can transform ex vivo and tissue explant models. Let's examine in the real application. I'm going to turn over to Dr. Albert Santamaria Martinez. Today, we're honored to have Dr. Albert Santamaria Martinez, who is a distinguished cancer researcher at EPFL in Lausanne, Switzerland. That Dr. Santamaria Martinez earned his PhD from the University of Barcelona in 2009 and conducted postdoctoral research in Professor Jörg Holzken's lab at EPFL, where his innovative work in oncology earned him the Pfizer Prize in Oncology in recognition in R. Weisberg, The Biology of Cancer. In 2014, he secured an SNF Ambition grant, leading an independent research group at the University of Freiburg, focusing on tumor microenvironment, cancer stem cells, and metastasis. Since 2020, he has worked in Professor Elisa Oricchio's lab at EPFL, developing lymphoma avatars from human tissues, a recent research published in Nature Communications. Dr. Santamaria Martinez, we will turn over to you.
Thank you, Christopher. This was a very generous introduction. And thank you, John, also for giving me the opportunity to participate in this very nice series of webinars by the Well Bioscience. So I'm going to be discussing in the following minutes these lymphomoids, which, as you may know, is a tissue-based ex vivo culture system that we developed in the lab for lymphoma therapy screening. And for those of you who are not familiarized with lymphoma, I would like to give a brief introduction. So lymphoma is a very heterogeneous group of tumors that arise from B cells that are blocked at different stages of differentiation. And I say B cells, although there are other types of lymphoma that arise from T cells or NK cells, but the main interest of the lab is non-Hodgkin B-cell lymphoma, which is what I'm going to be discussing mostly in this presentation. Now, non-Hodgkin B-cell lymphoma can be subdivided broadly from a clinical perspective into two big groups. So those tumors that are indolent and those tumors that are aggressive. In the indolent subtype, we can find lymphoma such as follicular lymphoma or a small lymphocytic lymphoma, while in the aggressive group, we may find tumors such as diffuse large B-cell lymphoma or Burkitt lymphoma. So as I was saying, there's these two groups of non-Hodgkin B-cell lymphoma, the aggressive and the indolent. And then one of the main features of lymphoma is that they rely heavily on the tumor microenvironment to grow. And this is particularly true in indolent subtypes of lymphoma and at early stages of lymphomagenesis. Of course, when tumor cells acquire more and more genetic and genomic alterations, they rely less on the tumor microenvironment to grow. That is why when it comes to model lymphoma, cell lines are useful to study aggressive subtypes of lymphoma or certain features of aggressive subtypes of lymphoma, but these are not available for early stage or indolent lymphomas. For example, there are almost no bona fide cell lines for follicular lymphoma. They are not useful either to study the tumor microenvironment, but on the bright side, they are cheap and fast to use, this we know. That's why some groups have developed 3D systems of lymphoma, such as these multicellular aggregates of lymphoma cells, and more recently, organoid-like structures of follicular lymphoma and DLBCL, but they still have trouble recapitulating the complexity of the tumor microenvironment. So a lot of the research in the lymphoma field is still done using mouse models, which are mostly based on transgenic, chimeric, and PDXs, so patient-derived xenografts, which, as you know, is time-consuming, expensive, and also poses some ethical concerns. Now, the question is, why is it important to model lymphoma? Well, it is important because of refractory disease and relapse, which are relatively common. And here I'm showing you just two subtypes of lymphoma, which account for more than 50% of all non-Hodgkin lymphoma cases. So non-Hodgkin B-cell lymphoma cases, which is DLBCL, so an aggressive subtype, as we said, and follicular lymphoma, an indolent subtype. And the DLBCL, so diffuse large B-cell lymphoma patients, which account for 30% of all non-Hodgkin B-cell lymphoma patients, out of these, 30 to 40% of these patients are refractory to first-line treatments or relapse within the first 24 months. And as for follicular lymphoma, so 20% of these patients that undergo treatment do not respond or relapse, at which point they have poor prognosis. And around 50% of the cases, so follicular lymphoma will eventually develop, transform into a more aggressive form of diffuse large B-cell lymphoma that requires treatment and is associated with poor prognosis. Now, the first-line treatment for many lymphoma subtypes is immunochemotherapy. So meaning a cocktail of chemotherapeutic agents combined with an antibody that targets molecules on the surface of, in this case, B cells, for example, CD20, so the antibody rituximab. But a second-line treatment for those patients that cannot receive, for whatever reason, chemotherapy, so we have a wide range of targeted therapies. For example, Bruton tyrosine kinase inhibitors such as ibrutinib or PI3 kinase inhibitors such as idelalisib. The question is basically that there is a need to establish knowledge-based protocols that help guiding treatment choices on a personalized basis in lymphoma patients. Or in other words, we need to tailor specific treatments to a particular individual, and for this, we need to know which treatment will be beneficial for which patient and which treatment we can spare the patient from. Now, in the next slide, I would just to show the current workflow is a very, very simplified workflow, but just how things work currently and then where we would like to come in. So let's say that a patient visits the doctor with a symptomatology, and then, of course, the doctor will run a series of tests and maybe, schedule the patient for a biopsy, tissue biopsy. Then this biopsy will go to pathology, and then there will be a diagnosis will be made upon the results of the pathology report and all the other analyses. And then the doctor will face the question on which treatment to give to the patient. This is currently based mostly on hospital guidelines. As I said, okay, first-line treatment, we know immunochemotherapy, but where we would like to come in, obviously, is here, where the biopsy, when the biopsy is performed, and then take part of this biopsy and in the lab, test the sensitivity to different treatments, and then inform the molecular tumor board or the clinician about which a particular treatment will benefit the patient most. So with this in mind, these are the aims that we had for this project, which were to generate a system to grow lymphoma tissues ex vivo, to use it as a diagnostic tool for personalized medicine, to study the biology of the tumor microenvironment in tumor explants, and to determine the spatiotemporal dynamics of tumor evolution associated with the acquisition of genomic alterations. So for the sake of time, I'm going to just be discussing the first three aims. And I'm going to start with the first one, which is how we generated this system to grow lymphoma tissues ex vivo. And of course, me coming from the cancer stem cell field, so it's a bit weird that I ended up working on lymphoma, if you want, which is widely believed not to contain cancer stem cells. So I thought that organoids would not be an option in this case because it's mostly based on the presence of cancer stem cells. So then I thought that maybe, since we need to maintain the tumor microenvironment, tissue explants would be a better approach. To do this, there is this one very nice paper published some years ago by the group of Neil and collaborators. It's the group of Calvin Kuo, in which they were using the air-liquid interface system to grow these organoid-like structures that contain part of the tumor immune microenvironment, mostly lymphocytes. So I thought that could be a good approach to test. Now, the air-liquid interface system is done, at least in this paper, using collagen as a matrix, which I tested. But for lymphoma, it didn't seem to grow to work so well, because I guess one of the reasons is because lymphoma do not contain, so these tumors do not contain a ton of collagen in the extracellular matrix. So that's when I found out about this VitroGel RGD. I have to, say I don't own stocks in the company, but I would just like to say a few words, because it has been very helpful. And I would just summarize the advantages that I encountered, using this hydrogel in 2 words, which would be the fact that it's ready to use and that is very fast. So it's ready to use. It means you can keep the bottle in the fridge, and then when you have to perform the experiment, you just take it out of the fridge, leave it at room temp or at 37 degrees, and then you will mix it with your media. You can fine-tune the strength of the gel, which is something that we did. I'm not going to discuss the details in this presentation, but it's a very fast process. And this is something that is very important in projects like this, because we try to keep the timing from obtaining the biopsy to seeding the biopsy as short as possible. It means that from the moment where the gesture is performed on the patient to the moment where we take this biopsy to the lab, we try to make it less than 10 minutes. And then, it's just mixing the components and seeding the tissue explants, which will take a bit longer, obviously, but it can be done very fast using a system like this, which works, as I said, pretty well. So this being said, how we did this was using a mouse model. So we started with a mouse model, which is this VAPI PCL2. This is a mouse model that forms tumors in the spleen and the lymph nodes that resemble follicular lymphoma in humans. So we thought it would be a good mouse model to do these tests. So what we did here was to take the spleen of these mice, chop them in different, in small fragments around less than 1.5 cubic centimeters, and then seed them in the air-liquid interface system using the VitroGel, and then look by histology at the architecture. We tested different conditions, obviously, on the gel and the different compounds that we add in the medium. And we found the right conditions that are reported in the manuscript to maintain the architecture, but also the viability and composition. This is something that we check by flow cytometry in time course type assays, looking at the percentage of viable cells and the percentage of different populations of cells that can be found in these tumors, such as B cells, and then germinal center B cells and T cells, different subtypes of T cells, and so on. And then by immunofluorescence staining, we looked also at the proliferation of these B cells and how we were able to maintain over time, the time courses of 3 to 5 weeks. We perform other types of assays, such as single-cell RNA sequencing. If you're interested, you can look at the paper. I'm not going to go into details because I think that is more interesting that we move to the human part. So I'm just going to conclude this part by saying that we developed this tissue culture system that maintains lymphoma cell growth and preserves the tissue architecture, microenvironment, and molecular profile of the parental tumor, and that due to the origin and nature of these explants, we decided to call them lymphomoids. So then we have this system ready. And now what we wanted as the second aim is to be able to potentially in the future use it as a diagnostic tool for personalized medicine. And here is a bit the workflow for human lymphomoid generation that we follow. So Every time that there is so that we recruit a patient, so the patient agrees and signs the consent, so there is a suspicion of lymphoma, and therefore he has to undergo these paraclinical exams, and then at some point the biopsy. If all conditions allow for it, we will take one piece of the biopsy, run to the lab, and then chop this biopsy in different, so in small fragments, and put them in this air liquid interface system using the VitroGel, and then put them in different wells that we will treat with different drugs individually, and then after the 3 to 7 days, we will run a series of, so we will fix the tissues, and we will run a series of downstream analyses, which are based mostly on multiplex immunofluorescence staining, with a panel that we designed specifically for lymphoma that includes CD20 for B cells, then CD8 and CD4 for T cells, CD68 for macrophages/histiocytes, and then Ki-67 to look at proliferation. And then, of course, what we want to do is to look at the response to therapies, or the sensitivity, to identify the most suitable therapy for each patient, and then, in addition, to perform some analysis of tumor structures with imaging and molecular technologies. So here's the lymphoma cohort that we had. So it's quite diverse. It represents a bit a wide spectrum of a non-Hodgkin B-cell lymphoma, and it includes actually one T-cell lymphoma. So you can see we got, obviously, most of them were diffuse large B-cell lymphoma and 5 primary mediastinal B-cell lymphomas. And this is how one of these biopsies, this is a big biopsy, looks like when we perform these multiplex immunofluorescence stainings. So for the different markers, it looks very colorful. Here the colors are splitted. If we look a little bit more in detail, so a bit of a close-up, of zoom in, you can see that it's very, as I said, a very colorful picture. It's confetti-like, but we can see all these proliferating cells. And then, of course, what we wanted to do here was to quantify these images. That's why we coded a computational pipeline to be able to analyze and digitalize and quantify these images. And then this is how they look, the image digitalization. Then we can quantify the number of B cells, of CD4, CD8 T cells and macrophages, and also the percentage of cells that are proliferating. In addition, obviously, that will give us the distances between cells and their position, so we can perform some neighborhood analysis. Now, this is how a representative picture of a lymphomoid looks at T0 and after 3 days in culture. This is an immunohistochemistry staining with Ki-67 in this case. By T0, obviously, what I mean is that the lymphomoid was produced from the biopsy and then fixed immediately. So it has not gone through cell culture. And so then here at your right, you will find the lymphomoid, how it looks like after 3 days in culture. As you can see, we can still find the proliferating cells. It looks pretty good. Of course, you cannot compare exactly the lymphomoids it's not a one-to-one comparison, because every time that you produce a lymphomoid, it will be different from the other. But you can see that the proliferation is there. And then if we look at cliff caspase 3 staining, so as we report in the paper, there's a little bit of cell death, of apoptosis going on, especially in the corners. But overall, the viability and the proliferation of the lymphomoids are maintained quite well. Obviously, this is something that we did, systematically by comparing the biopsy to the lymphomoids using this multiplex immunofluorescence staining. And then if we look at the different markers that we use, CD20, 4, 8, and 68, we can see that there is a strong correlation for all markers between what we see in the lymphomoids and what we see in the tumors. and also in the percentage of proliferating B cells that we find. Of course, what we wanted to do next was to test different targeted therapies in these lymphomoids. And this is something that we did using compounds that are typically used in the clinic, as I said, as second-line treatments. We had a panel of drugs that we used on these lymphomoids. What we saw is that some tumors would respond to some therapies, but not others. Some tumors would respond to nothing. Some tumors would respond to everything. So it was a bit diverse, which was, I think it was very interesting and somehow what we were hoping to see. Obviously, there's the thing that I will discuss in a couple of slides, which is that, of course, what you want then to see is whether there is a match between what you see in your explant cultures and what happens in the clinic. But I would like first to show you two more slides, which are the following. First, that the sensitivity response timing, we tested 3 days versus 7 days. And it is very similar. So basically, those tumors that will respond to a certain therapy at 3 days, they will respond also at 7 days. And those that they don't respond at 3 days, they typically don't respond at 7 days. So what we see in some cases is that at 7 days, of course, there's a bit of decrease in the overall viability of these lymphomoids. You have to think that the tissue explants is not a system that that is like the organoids that you can passage forever and so on. It's a system that it's short-lived, so it's meant to be cultured for a few days to a few weeks, and also that our aim is to report to the molecular tumor board. So this report cannot take too long. So we think that 3 to 7 days is a good time window to give the report. The second thing is, obviously, we chose CD20 as a marker of B cells, but also, as a proxy for tumor. And we had the question of whether this was actually good enough, because obviously CD20 will stain cells that are not tumor, that are not transformed. So we did an additional staining, which is the light chain staining, which is a molecule that is expressed specifically by B cells. And so in lymphoma, it is used to see clonal expansion. So we have the lambda light chain and the kappa light chain. And we knew from the pathology reports each tumor which clone it was. So for those tumors that we had clinical data, we run these immunohistochemistry stainings, we quantify them, and what we saw in the lymphomoids, and what we saw is that indeed it was showing the same trend that what we would see for CD20, which was validating our approach of using CD20 positive, Ki67 positive cells as a marker for sensitivity to the different targeted therapies that we used. So finally, what I was saying is that, of course, what you want to see in a project like this is that what you see in the culture is the same that the clinician will see in the clinic. Now, this is quite challenging in a project like this, because this is a proof of concept project. Plus, as I said, most patients, first-line treatment, they will receive immunochemotherapy. And this is not something that we tested for different reasons. But in 7 cases, so in 7 patients, and it's 9 cases because 2 patients had to, were given 2 different therapies because they were not responding. We have this data on how they did, whether they responded to treatments that we actually used in our tissue explants. And in 8 out of 9 cases, we were able to predict correctly the outcome of the patient. And I'm just showing here 3 patients, 5 different examples, including the one that we, did not predict correctly. So just to be fair. And then you can see, for example, this patient that suffered from a small lymphocytic lymphoma. It was given a BTK inhibitor, acalabrutinib, and then was showing on the PET scan a complete, a metabolic response, while in our explant culture system, another BTK inhibitor, ibrutinib, was also showing a strong inhibitory effect. So overall, we can conclude that the lymphomoids can support the viability and composition of human lymphoma tissues ex vivo, and they may be used as a tool to predict sensitivity to target therapies in lymphoma patients. I'm saying maybe because obviously, this cohort needs to be much bigger. This is something in which we are working now. Finally, I will use these last slides to show you basically the studies of on the biology of the tumor microenvironment in these tumor explants that we did and we're currently doing. And we started by using spatial transcriptomics. And this is the Visium by 10x. I'm sure that many of you are familiarized with it, but for those of you who are not, it's basically has spots of, if I remember correctly, 55 micron in diameter, that contains some 5,000 probes, and then you will hybridize your tissue with this. And then, of course, since these spots are 55 micron in diameter, it means that in our case, each spot may contain around 20 to 30 cells. So the information that you get from these spots needs to be deconvolved. So we applied base prism deconvolution here to understand the cell enrichment that we had in each spot. And as you can see here, for example, the clustering that our bioinformatician did, cluster here 5, you can see that it's enriched in tumor, and it was matching quite well to the annotations that our pathologist did on the H&E of this tumor biopsy. This is, by the way, DLBCL. So you can see that there's quite, we have quite a bit of heterogeneity here. And what I thought that it was very nice to see is that this heterogeneity is maintained when we looked at the, by spatial transcriptomics at these tissue explants, so at the lymphomoids. The second thing that we thought that it was interesting is that the tumor spots are still maintained. So because, of course, if we would have lost the tumor during cell culture, that would have been too bad. But we see one heterogeneity, which is good because anyway, we are randomizing these tissue fragments. And the second is that we see the tumor spots. Now, what happens when we look at the treatments? So this is a tumor that when we perform the multiplex immunofluorescence staining using, so on the lymphomoids that we treated with different compounds, we saw that it was responding to ibrutinib, but not to idelalisib. So ibrutinib being the BTK inhibitor, idelalisib a PI3 kinase inhibitor. And this is something that we saw equally well in the spatial transcriptomics analysis in the Visium, because here you can see that in the ibrutinib treated, so they are completely devoid of tumor clusters, while idelalisib treated lymphomoids, you still can see some of these clusters. This can be also spotted quite nicely here or in the spot compositions where the control looks similar to the parental tumor with the tumor here in brown. The ibrutinib is completely or almost completely gone, the tumor spot, while other populations of cells such as macrophages, endothelial cells, and T cells seem to be expanding. Idelalisib treated, as you can see, of course, here you can still see the tumor spots. So From this, we can conclude that spatial transcriptomics confirm that these lymphomoids are representative of the original tumor, that are sensitive to certain targeted therapies, and display distinct changes in the tumor microenvironment linked to specific treatments. And with this, I would like to finish. I would like to thank the people, so the team that made this possible. In particular, Justine Epinay was awesome. Did a great job, in particular with the patients, funding agencies, and you for joining this webinar. I'm happy to take questions.
Thank you very much, Andres, for your presentation. We have one more poll to go, and we'll proceed to your Q&A session, but keep them coming. In what ways can lymphomoids be integrated into clinical trials to enhance personalized medicine approaches?
I'm trying to do now first is to test the predictive power of the system by having a cohort of patients in which we can compare the treatment that they will receive. We will know the treatment that they will receive, and then apply it in the tissue explants, so in the lymphomoids. That's the first step. So we need to know exactly. So okay, here we calculated it's 8 out of 9, but these are very low numbers. So we cannot derive any, very strong, and we cannot make strong statements on this. So that's the first step. We need to make this code, and then afterwards we could start a clinical trial. Yes.
All right. I think some of these questions are kind of tied in, so I think I'm just going to might be repeatable. How does the ex vivo model compare to patient-derived PDX models? And what are the advantages, disadvantages in terms of experimental duration, preservation of tumor architecture, and microenvironmental interactions?
Mm-hmm. Okay, so in terms of timing and efficiency, they will compare pretty well, because xenografts, first off, they will take quite a while to grow. So to graft, those that graft, which is typically not such a high percentage. That's for starters. Then obviously those indolent subtypes of lymphoma, forget it. It will be almost impossible to make these grafts. Then obviously the tumor microenvironment will also be replaced by the microenvironment of the mouse. And that is not interesting, right? So to be used as tools for personalized medicine, in the case of lymphoma, I'm not so sure.
Can this lymphomoid system use patient biopsy be used also as a diagnostic tool in parallel with drug response recommendations? Basically something one step further from basic histopathology, but yet not too expensive, which can be paid by insurance.
What I can say is that what we tested in parallel was flow cytometry, which is also done routinely in pathology labs, right? If you don't want to do histology, one could do flow cytometry. The problem with flow cytometry is that then you will use the whole lymphomoid. So you will have one shot at making the claim of whether it's sensitive or not. Then afterwards, the sample is gone, so you don't have it. And like this, so since what we do is to look at different lymphomoids and at different depths of sectioning depths of the paraffin block, then you can get more data out of it, and you can use it for different purposes. Whether this type of assay would be covered by an insurance, that I do not know.
A question also, have you tested the VitroGel recovery solution? Do you think it could make long-term culture possible?
With what solution?
VitroGel recovery solution. It is one of our products that actually recovers cells from the hydrogel. Yes.
What I like better is to fix the whole gel so that I do not disturb these explants. Because there is a difference between these explants that come from lymphoma and other types of explants that you may have, or organoids that you may have from other tumor types, such as colorectal cancer, right? Those are more solid and more compact. And these are, very dynamic structures in which you may have cells that move a bit around. So dissolving the gel, you may lose some of the cells that you have in there. Curiosity, so the maximum that I have cultured these explants for is 10 weeks. And they were still okay. The mouse, that's the mouse lymphomas, the murine lymphomas. But I mean, for the purpose that we have, it's a trick that you can play in the lab. It's interesting to see, but not for the purpose of personalized medicine.
How did the stiffness of the hydrogel, the ability to tune it, affect your lymphoma tissue research? Did that have a play in that?
So
how did the stiffness of the VitroGel hydrogel, the ability to tune it, affect your lymphoma research?
Yes. Okay, so we try to find a compromise. So I try to find a compromise between so the gel being too soft and the gel being too hard. So what we saw is that gels that are too hard, then cells tend to die. So what happens is that, which has been shown by a couple of groups, at least I think, that then you activate CD8 T cells, and then CD8 T cells will go kill the cells. It's a rather interesting phenomenon. But they don't like it. They certainly don't like very strong gels. Now, if the gel is too soft, what will happen is that then you cannot contain the explant, and then the cells start making kind of a cloud, which you don't want either, right? Because then the interactions between the different cell types may change. So I did quite a few testing on this, and we found that this gel strength was the best for this purpose. I'm not saying for every purpose.
Perfect. I think we might have one last question left. What are your thoughts on the B-cell lymphoma explant, lymphomoids showing decreased viability after 72 hours? Is there a change in the cell status or phenotype?
So after 72 hours, there is a small decrease in viability that will become probably more acute after. So the longer you culture this until it becomes, so there is a plateau that it will be reached. What we have observed is that the different populations, so there are no populations of cells that seem to be more sensitive to these. So they will all die equally, and this will be most likely due to the process of, obviously taking this sample out of a human being and putting them in some exogenous conditions. And this happens with anything that you do, that you take out of a living being, right, and you put it in cell culture, especially if you want to maintain architecture, the composition, and so on, you will have a bit of this at the beginning for sure.
All right. That was our last question. We have a lot of other webinars online as well and upcoming as well on our explant models. We do have one in March 12th, 11th a.m. Please look out for that. And thank you, Dr. Artemis, and thank you, for that great presentation on using the explant models for your lymphomoids. If you have any questions, please follow us on the Well Bioscience, and also you can connect with Dr. Santa Maria Martinez as well. His information is there about his research and his paper as well that was recently published. It's on our website as well. On the webinar, we urge you to read his really interesting paper, really informative. For explant models in the future of medicine as well. If you have any questions, please just email us at the Well Bioscience at support@thewellbio for your explant tissue, explant models, or any of the 3D cell culture needs. Thank you, everyone, and have a great day. Thank you, Dr. Martinez, for your presentation. Have a great day, everyone. Thank you.